And so the next speaker is Professor Sir Ravinder Maini from the Kennedy Institute at Imperial College of Medicine in London. As a clinician scientist, Professor Maini and his colleagues have been instrumental in bringing the findings of basic science into the clinic, particularly in the field of rheumatoid arthritis. Today he's going to talk about anti-TNF monoclonal therapy, a journey from a concept to a standard of care in rheumatoid arthritis. Welcome.
Chairman, ladies and gentlemen, first of all, thank you to John Dirks and the Gairdner Foundation for this marvelous symposium, which I have greatly enjoyed, learned a lot from it, and wonderful to see how this field is progressing and the possibilities are opening. Well, today I shall be covering a bench-to-bedside story which took 15 years, roughly. And it's an important story because I believe it's the first instance where a monoclonal antibody was used for a chronic disease and was able to be used continuously and set a standard for therapeutics in medicine. Okay, so first of all, like everyone else here, I'd like to congratulate Greg, whose technology has hugely contributed to this field, especially the possibility of using antibodies for prolonged periods. I mean, to my knowledge, patients on anti-TNF antibodies have now reached at least the 10-year mark, and some of them 15-year mark, because as you probably know, this is continuously needed. So these really are established, robust therapeutics. And the other important point about this slide is that at least 4 of these antibodies in each class of the process of humanization are anti-TNF antibodies. And I think we've probably heard more than one person refer to the fact that anti-TNF agents, which includes the soluble receptor IgG fusion protein, occupy a huge space in terms of commercial success. I believe in 2012, $25 billion turnover was attributed to anti-TNF agents alone. So obviously the therapeutic commercial success has been a huge driver in this field. Now, at the outset, I'd like to thank and acknowledge my partnership with Mark Feldman, without whom this work would not have been possible. It certainly would not have been very enjoyable or very fruitful. And I think that the synergies between our expertise were very instrumental in this project being driven to its conclusion. We owe a huge debt of gratitude, not only to the team at the Kennedy Institute, and in these 15 years, there must have been at least a dozen fellows who went through our program, 4 or 5 technicians, at least 2 or 3 nurses, countless collaborating clinical units in Europe and North America, and of course, a very interesting collaboration with biotech industry, which had its moments of difficulty, but actually was conclusively, in the end, demonstrating the rule that this kind of work really is not possible in academia alone. It does require collaboration with industry. So I'm going to cover the first 5 years' work in very, very brief 2 slides. But before I do that, let me just tell you a little bit about the disease that we're discussing today, namely rheumatoid arthritis. This is a very common disease. 1% of the population worldwide suffers from it. It is a disease which is painful, and the pain in initial stages is due to inflammation, but relentlessly progressive in the days that we started this story was the underlying destruction of cartilage and bone of joints. And that really is the cause of disability over time. And that is irreversible, whereas inflammation, to a certain extent, was controllable already in the mid-1980s and 1990 when we started this project. Now, I think you'll be interested to see on the left-hand side the pathology of rheumatoid arthritis, which explains the inflammatory component from the structural damage component. The inflammatory component basically is in a chronic immune-mediated reaction in the synovial tissue, which is the tissue lining the joint, and it is characterized by infiltration by hematopoietic-derived immune and inflammatory cells, and some proliferation of local connective tissue cells. There is an inflammatory exudate enriched with polymorphs and lymphocytes in the joints, which causes the swelling. These two characteristics are responsible for the pain and swelling and stiffness that the joints experience and leads to immobility. And then here you see a finger joint, and what you see is destruction of cartilage and bone. Here's a bone erosion, which really, in the end, is the part of the disease which really is only treatable by surgical intervention and joint replacement. Now, the tumor necrosis factor is a member of the family of cytokines, which act as intercellular signaling molecules. It acts by binding to one of its two receptors. There's a type 1 receptor and a type 2 receptor. And in rheumatoid arthritis, TNF-alpha is predominantly produced in the joints by macrophages. The receptors are widely distributed, especially the type 1 receptor, which is found on practically every cell type in the environment of the macrophage. And because this is a cytokine which acts mostly in its local environment, you can see why it is necessary for the ligand and the receptor, to be actually geographically co-located, which it is in rheumatoid arthritis. Now, I should explain at this point, because people often ask me this question, why is it called tumor necrosis factor? And the reason is that its discovery was related to the action of killing of tumors of this cytokine in experimental models. But in humans, this proved impossible because the dose of tumor necrosis factor, which might just be toxic to cells, is too high for the body to tolerate, because basically it leads to a syndrome of shock. Blood pressure goes down, temperatures go up. And volunteers who received this in the early stages, when the recombinant TNF became available, it became quite obvious that this could not be given as a systemic therapy. It was for a time used as regional perfusion therapy of tumors, especially of sarcomas, with only moderate success, so it was abandoned. Now, from the point of view of our story, it was access to cDNA and antibody probes that allowed investigation of tissues, which is where this game started, and without which we would not have been able to investigate expression of TNF in tissues of patients and controls. Now, what turned out in the initial phase was actually quite depressing, because we discovered that, in fact, multiple cytokines were expressed in rheumatoid joint. You name it, and it was expressed. So every reagent that we could get hold of at that time, between 1986 and 1990, TNF, IL-1, IL-2, IL-6, chemointerferon, GM-CSF, and so forth, every one of them was expressed, some in more abundance than others, but you could not get away with the fact that they were all there. Each of them might be doing something very interesting, very important, but there were, of course, overlapping functions, and the whole story was depressing, because it seemed, from the point of view of therapeutics, that no single cytokine was likely to be an ideal candidate as a therapeutic target. Well then, an experiment conducted in Mark's laboratory by Fionnuala Brennan essentially gave us the first clue that the cytokines expressed in rheumatoid joints were not acting simply in random and in parallel, but they were linked in some kind of a network. And the experiment depended on the fact that if you take rheumatoid synovial joint cells, disaggregate the membrane and then culture the cells in tissue culture medium, then for up to a week, they replicate exactly what's going on in the body. They produce the same range of cytokines in the same sort of concentration that we're observing in vivo. And in this system, if you add an inhibitor, then you can ask and answer the question whether any one of them is regulating any other cytokine. And the only inhibitor that appeared to do that was a neutralizing anti-TNF antibody, and which markedly reduced the production of interleukin-1, and subsequently we discovered it also downregulated interleukin-6, GM-CSF, and interleukin-8. So that gave us the clue that there was some kind of a hierarchy or a cytokine network of some sort operating in the joint. Well, it was a simplistic idea. It's much more complicated today. I don't have time to go into that. But the second experiment, which was conducted in my laboratory, was conducted with a monoclonal anti-TNF neutralizing antibody in a mouse model of rheumatoid arthritis. And basically what it showed was that after the onset of rheumatoid arthritis in these mice, or rheumatoid arthritis-like disease, the anti-TNF was markedly inhibitory for both suppressing inflammation and, more importantly, suppressing joint damage. Well, so by 1991, we were ready to ask ourselves the question whether this was true in humans. And again, to cut a long story short, it was quite difficult to persuade the pharmaceutical and biotechnology industry that had anti-TNF antibodies to allow us to experiment with their inhibitor in man. But it so happens that in the late 1980s and early 1990s, Sarami's work had led to the conclusion that septic shock was driven by TNF. Again, it caused cytokine storm, multi-organ failure, and at least in animal model systems, giving these animals an anti-TNF antibody at the outset of the septic shock led to a complete amelioration of the shock syndrome. So many companies and biotechnology companies developed anti-TNF agents. And at the NYU, Jan Wilczek developed a mouse, high-affinity mouse anti-TNF specific antibody. And Centocor, which was a biotech company, acquired this antibody. Their scientist, Jim Gareb, humanized it or chimerized it, I think, using Greg's technology. And we were very lucky that Jim Woody, who was the chief scientist at Centocor, who knew us, actually was one of the few people who was not very skeptical of our work and agreed to allow us to treat initially 10 patients, but subsequently full 20 patients with this chimeric anti-TNF alpha specific antibody. Now, we were naturally quite worried about doing this work. There was considerable skepticism that neutralizing TNF alone in a human physiological system in disease, in which many cytokines obviously were playing an important part, could have a profound effect. And moreover, no monoclonal antibody to date had been used for any long-term therapy. And it was quite clear that chimeric antibodies were likely to be immunogenic, even though only about 1/4 of the antibody molecule was human, or was of mouse origin. There were also safety and cost concerns, and I think at that time people believed that patients would not tolerate long-term therapy with injectables. Well, the first 20 patients was essentially a safety trial, but also a biomarker trial. And what we discovered was that in these 20 patients, 18 of them showed a very remarkable, what you might call dramatic response to the antibody injection. 20 milligrams per kilogram injection was injected, which is a massive amount of antibody, but from our animal work, we knew that quite a lot of antibody would be needed for the amount of TNF that was being produced in the 68 joints of the patient that were inflamed. So there was this remarkable clinical improvement, and at least in the short term, there were absolutely no safety signals. And indeed, the patients were sitting up in bed asking us, why you're so worried? Why do you have a nurse here 24 hours a day? I'm feeling fine. We discovered from this initial study that something quite important, which was that the duration of the response paralleled the blood level of the terminal part of the response period. So when the blood levels approached 1 to 3 micrograms per mil, the disease came back. So it was obvious that this was not a cure for rheumatoid arthritis. Well, it was necessary then to perform a placebo-controlled randomized trial where the patient, the doctors, and the observers were blinded to the treatment. And what you see here is that the two doses that were used and compared with placebo, at the high dose, 80% roughly of patients responded at 4 weeks. At 1 milligram per kilogram, 44% responded. And with placebo, only 8% responded. A very clear signal of efficacy. But of course, since rheumatoid arthritis doesn't last just 4 weeks, the question was, how long would this benefit last? And we were fortunate enough to be able to retreat 8 of the original 20 patients. And what you see here is a pretty typical graph. And you see that the patient received initially a rather big dose and then subsequently half the dose. And what we are plotting here are the swollen joint counts and the C-reactive protein, which is a marker of inflammation. And you see a rapid, very rapid response, but after about 10 weeks, the disease comes back. Another shot, rapid response, 10 weeks or so, disease comes back. Now, this last time when the injection is given, there is a shortening of the response period. And this patient, like 6 of the 8 patients, had developed an anti-chimeric antibody. And so we knew that the impact on the disease magnitude was not impaired, but the duration of the response was getting less. And there was a dramatic example of a patient who, on grounds of special need, was allowed to have an unlimited supply of anti-TNF injections. And in this particular patient, the anti-drug response was so profound that at the last injection, the benefit only lasted 3 days. So it was very obvious that this immunogenic antibody could be troublesome for long-term therapy. And the question was, well, what could we do about it? Well, in those days, there were no fully human or even partially humanized antibodies. That came much later. So we went back to the laboratory and worked with the animal model and came up with the finding that combining anti-TNF with so-called immunomodulating agents led to synergy. And to cut a long story short, a drug called methotrexate was the gold standard drug used for the treatment of rheumatoid arthritis in man. And this drug is an immunomodulator, as well as being a very profound anti-inflammatory agent. It has multiple actions, and we don't really know what the real important action is in rheumatoid arthritis. Being empirical, we didn't worry about it too much. We said, well, let's combine these 2 drugs and see what happens. And basically, we discovered something very interesting. So this was a study in which combination therapy was compared with monotherapy. And you see that at the 1 milligram dose, there was a rapid desensitization. response was lost by 8 weeks in every patient in this group. There were 23 patients in this group. Every one of them had no response by the time of 6 weeks. But the patients that had co-therapy with methotrexate showed a response period which really went well beyond the last injection at 14 weeks. And you see the similar kind of data for the terminal part of the combination therapy with the higher doses of antibody, although you also will observe that the antibody alone at the higher doses appeared to work much, much better than at the 1 milligram dose. And the reason is shown there. So you see the incidence of Remicade or infliximab antibodies was more than 50% in the patients that received anti-TNF without methotrexate. At the 3 milligram, it was less, and at the 10 milligram dose, it was virtually absent. Whereas in combination with methotrexate, this was heavily suppressed at all doses. Now, this is a very interesting phenomenon. We believe it is due to induction of high dose tolerance. And indeed, if you give this type of chimeric antibody so that the blood level never decreases beyond 3 micrograms per mil, the patient will tolerate it for a long period of time. And that turns out to be true for all other potentially immunogenic immunoglobulins that have been used in the clinic. So methotrexate combination was very interesting, but although it's partly explained by the phenomenon on suppression of HECA, that isn't the sole explanation. We believe there are other reasons for this, and the synergy probably involves modulation of other molecules in the cell. Well, the key phase 3 trial lasted 102 weeks, and you see there are 3 doses being used of anti-TNF with methotrexate compared with methotrexate alone, and you see at the end of 2 years, patients on the combination therapy were doing fine compared to methotrexate alone. And more importantly, these are the radiographic scores which measure the amount of damage to joints. And what you observe here is that over the 2-year period, patients on methotrexate alone, the joint damage progressed in a linear fashion, but patients who received anti-TNF plus methotrexate, it virtually completely suppressed progression of joint damage. So that sets the scene for the co-therapy being the standard of care. And basically, you start patients on methotrexate to which 50% will respond, and the 50% that do not respond are candidates for anti-TNF therapy. And in these patients, there's rapid and marked improvement in symptoms, signs, and laboratory biomarkers. In early disease, you can discontinue the antibody after about a year, and it can continue to be maintained on methotrexate alone. But this is virtually never possible if the disease has been present for more than 1 to 2 years. So the most important part of this study is the massive and impressive protection of joint cartilage and bone and tendons from deformities, and therefore maintenance of physical function. And this is reflected in better quality of life, and interestingly enough, reduced comorbidity, cardiovascular disease, and no wheelchairs, less surgery. Now, you see here, the 2 first anti-TNF agents were infliximab, which is the antibody we worked with, and etanercept, which is a type 2 receptor IgG fusion protein made by Amgen, Immunex taken over by Amgen. Now, this therapeutic inhibits not only TNF, but also lymphotoxin. And it may be the reason that a small proportion of patients, I would say hardly 5% of rheumatoid patients, that do not have a primary response to the antibody-based therapy, which is specific for TNF-alpha alone, may respond to etanercept. But otherwise, the efficacy profile is virtually identical for all the anti-TNF agents. And you see there are now 5 in the clinic, and earlier this summer, one biosimilar has been licensed for use by the EU regulatory authorities, but not yet anywhere else, except in Korea. Well, serious adverse events are a feature. Fortunately, they are rare. And if you select your patients carefully, they're almost not there. So we can generalize and say that in well-regulated circles, this is a very well-tolerated disease. When serious adverse events occur, they tend to occur within the first 6 months. And the most important of these is an increase in serious infections, particularly intracellular infections, such as reactivation of tuberculosis. And this is a serious problem in parts of the world where TB is endemic, and people basically control their disease, but the organism is still living. And if you give these patients anti-TNF without prior screening and without prior treatment of the so-called latent tuberculosis, you're in trouble. But people have now learned what to do. All clinics will very carefully scrutinize and examine patients for so-called latent TB, and if they suspect it's there, then they will be treated with a course of anti-TB drugs simultaneously with starting anti-TNF therapy. And that has made the incidence of TB virtually non-existent. But other infections do occur. For example, staphylococcal infections, pneumonias, these do occur. And you have to be alert because patients can become very sick very quickly because the anti-TNF is suppressing their inflammatory response to the infection. And they may turn a corner very quickly into being seriously ill. So the initial clinical trials suggested there was an increase in B-cell lymphomas. Now, it turns out that in rheumatoid arthritis of the severity that was present in the majority of patients that we treated in our early studies, actually had a 28-fold increase in the incidence of lymphomas, which is related to the severity and duration of rheumatoid arthritis. And that is because rheumatoid arthritis itself is immunosuppressive. It suppresses the immune system in an attempt to suppress the disease, but it overshoots in the suppressive aspects of the response to disease. And we believe that is the reason why lymphomas might be so much more common. And in fact, this is now borne out by registries, long-term follow-ups in USA, Britain, and Sweden, where it turns out that patients who went into trials later than the early trials or in practice, actually there is no increase in lymphoma signal, except in children. So this hasn't gone away. There is still a slight concern that in certain populations, lymphomas might occur. Then the other thing that has turned out is that certain forms of skin cancers are more common in patients receiving long-term anti-TNF therapy. Now then, how does anti-TNF work? And I'm going to spend just a few moments telling you about that. It inhibits IL-6 production. It inhibits C-selectin and VCAM, cell adhesion molecules. It inhibits IL-8 and MCP1, two important chemokines. It inhibits VEGF, which is a potent angiogenic factor. It inhibits metalloproteinases, which are involved in protein and bone destruction. And interestingly, it restores immune homeostasis. Now, I won't have time to discuss this in detail, but here is our study in the regulation of IL-6 after anti-TNF. And what you see here is that within 1 day, there's a suppression of IL-6 production after a single injection of anti-TNF antibody. And this is telling us that the anti-TNF, which is injected into the bloodstream, is entering the joint, neutralizing TNF in the joint, down-regulating IL-6 production in the joint, which is in turn down-regulating CRP production, by the hepatocyte in the liver. So it has a widespread systemic effect as well as a local effect. And in fact, recent findings have confirmed something we suspected at the very beginning, which is the feel-good factor that anti-TNF gives patients. Patients tell you that they feel great. And we always suspected there might be some effect on the brain. And recent studies on the brain using fMRI suggest indeed this is the case, that parts of the brain which show abnormal activity in rheumatoid patients before treatment become altered to normality in these people. So there is a CNS effect, which is quite early on after the injection of anti-TNF. This is the second major effect of anti-TNF therapy, which we discovered very early on, because actually it was quite difficult to re-biopsy joints of patients. So this was a study in which we were biopsying a knee joint before and after anti-TNF therapy, 2 weeks after. And we actually found it very difficult to re-biopsy these joints because they had dried up in the meantime. And you can see what had happened. And here was the pre-treatment cellularity, plenty of macrophages, plenty of CD3 positive T cells. Here's the rather rudimentary looking synovial membrane after only 2 weeks of treatment. And you see a great reduction in CD68 positive macrophages and a marked reduction in CD3 cells. So basically, what is happening is that the down-regulation of adhesion molecules and chemokines is leading to blockade of recruitment of cells into the joint. And here's a direct proof of it. In this study, Peter Taylor, who was then my research fellow, labeled white cells with indium-111. He imaged the joint before anti-TNF therapy. Here are the knees, here are the wrists and fingers, and you see plenty of uptake of isotope because this is where the labeled cells have become trapped. After anti-TNF, the experiment is repeated, and you see virtually abolition of the trafficking of joints into inflamed cells. Well, I've already alluded to cartilage bone protection, normalization of T regulatory cell function, improvement of effector cell function. But these are temporary effects. Otherwise, you'd expect the disease to actually go into long-term remission. And the question is whether early in the disease, when the disease is not yet very complex in terms of antigen spreading, whether at that stage of the disease it's more amenable to being arrested. And it seems that might be the case, and maybe this is the underlying factor, although that hasn't been proven. B cells also become less active, macrophage activity and cytokine activity production is reduced. Now this work, which started in rheumatoid arthritis, obviously engineered a lot of other interesting questions from other inflammatory diseases, and it has now been shown and is widely used as treatment for juvenile chronic arthritis, ankylosing spondylitis, psoriatic arthritis, psoriasis, Crohn's disease, and ulcerative colitis. The success of anti-TNF has opened floodgates of therapeutics using antibodies. It has had a transformational effect in encouraging development of mAb-based therapies. In the clinic, there are already several other non-anti-TNF biological agents, an IL-1 inhibitor, a CD20, anti-CD20 B-cell antibody, a CTLA-4 Ig, which inhibits CD28 interaction with antigen-presenting cells, and an anti-IL-6 receptor antibody. In the pipeline are at least 2 other anti-IL-6 antibodies, an anti-GM-CSF antibody, and 2 anti-IL-17 antibodies. Now, we await with interest to see what the results of this are. In fact, what has happened is that rheumatoid arthritis has become a test bed because there is such a huge amount of experience with these therapeutics in rheumatoid arthritis, that any company that develops a new anti-inflammatory immune modulating agent will try and experiment on rheumatoid to prove whether it's going to be a viable drug. And it has become a very overcrowded field. It's almost impossible in the UK to find new patients for trials, so the trials are now being done in deeper parts of Asia and Africa. And I think the results are becoming quite difficult to interpret because these are different populations of patients. And so I'm always a bit skeptical of the results we see at the present time. There are a lot of challenges, as I think demonstrated, I hope very clearly, that although the response is magnificent, it's only so in about 50% of patients. Over time, beyond 5 years, 50% of patients that responded originally will have dropped out for various reasons, usually due to some kind of a tachyphylaxis. In other words, they develop some kind of a resistance to anti-TNF therapy, which the others don't. Now, what distinguishes these patients from one another is an interesting and important question, and a lot of money is being spent on trying to identify biomarkers that might tell us what the differences are. I'm afraid that although at the group level, multiple biomarkers especially tell you something, there is no test available which will predict an individual patient's response. So this field is still in its infancy. It is not yet ready for use in the clinic. Cure remains elusive. This disease still requires new approaches, and there's so much more activity going on. I think people in this room probably are aware. People are interested in microRNAs, they're interested in toll receptors, in inflammasomes. There really are potentially other very interesting targets, which is making this a lively field. And if I was 25 years younger, I'd be out there in the front working and trying to make some progress. Sadly, I still have to watch from the back seat. Thank you very much.